Good day everyone, good day Mrs. Flores. I am AJ Vincent A. Bacdat and I'm going to present to you the DNA replication. Now the DNA replication, it is the biochemical process in which the DNA unwinds itself and produces exact copy of themselves. In an overview, the DNA starts with the two strands of the DNA as represented by this zipper because it is somewhat looks like an operating zipper when it starts the process of unwinding, just like this one. Now we have here the separated strands of the DNA, and each of these strands will serve as the template of the DNA replication. The DNA replication follows the complementary base pairing rule. It means that in every adenine, there is a presence of thymine, and in every cytosine, there is a presence of guanine. Here in my model, I have here the nucleotide as represented by this one. The brown one represents the phosphate, the white one represents the sugar, and the blue one represented the adenine. While here, the yellow one represented the thymine, the green one represented the guanine, the pink one, lastly, represents the cytosine. Now, there is three modes of DNA replication: the semi-conservative, conservative, and the dispersive. Right now, I'm going to present to you the semi-conservative mode of replication. It means that in every old strand, there is a corresponding new strand. Now, So here is our DNA replication model. To begin with, an enzyme called DNA helicase, which is this, will run along the parent DNA, or it will bind to the dimers, which will break down the hydrogen bonds between the molecules, resulting to unwinding of the DNA strands, forming a zipper-like structure and an opening structure we call the replication fork. So here are the procedure. So as the helicase runs through the middle of the DNA strands, the unwinding occurs. Which now the replication fork will be now visible. So here is our replication fork as shown in this diagram. So here is our replication fork. So after the DNA unwinds, the single-stranded binding protein will attach to the end of the old strand so that it will not flip back to its original form. Okay, so as you can see here, it will attach here. Okay. Now, the unwinding of the DNA strands occurs at any sites of the DNA. It runs simultaneously and it flows in bidirectional. Meaning, if we start here from left to right, it may also occur from right to left because it occurs in bidirectional model. Now, there is another consideration. The DNA polymerase can operate only into 5' to 3' ends. Now, in our model, the top of the old strand, which is this, started at 3' direction. So it means the end of the strands will be 5'. Okay? And then the old strand, the lower old strand, as follow the opposite orientation of the DNA, it will started at 5' to 3' direction. Therefore, the new strands, represented by this black zipper, the new strand on the top will run from 5' to 3' end. And then the bottom will be 3' and the end of it will be 5'. And now we have the leading strand and the lagging strand. The difference between the two is that the leading strand replicates continuously along the DNA strands and it follows the direction of the replication fork. Whereas the lagging strand as it starts with 3' end to 5' end, it replicates in an opposite directions, and it calls the lagging strand to unwind in segments. Remember the word segments because it will be shown later. And now, to begin with, we have now the DNA polymerase 3, which is this one, the DNA polymerase 3. And due to the absences of its hydroxyl group, they cannot bind yet. Therefore, the RNA primase will synthesize primer to the, which are complementary to the bases, and it will mark as the starting point of the construction of the new strand of the DNA. So let's attach the primer here at the leading strand. And since the lagging strand will start from right to left, because again, it follows the 5' to 3' direction, therefore the primer will be attached near to the replication fork. As you can see, the helicase starts to break down the DNA strand through the binding of the thymine and adenine. Why is that so? Because the bonds between the thymine and the adenine is composed of double bonds as compared with the guanine and cytosine that is bonded by 3 bonds. Therefore, it is easier for the helicase to destroy or to break down the hydrogen bonds between the adenine and thymine as compared to the triple bonds in guanine and cytosine. So now, let's proceed. As we put the primer here and the new strands, we may now add our nucleotides. So let's follow the direction of the replication. So in our lagging strand, we have here the guanine, therefore it will be paired by cytosine. Okay, let's thymine by adenine, cytosine by guanine. Let's spread it pa. Let's spread this one so you will be able to see. Okay. And now let's now proceed in the lagging strand, which starts from right to left. So as we can see here, we have here the adenine, so it will be paired by thymine, and then guanine to cytosine, and thymine to adenine. Now, as you can see, as we proceed to the DNA replication, the helicase continuously breaks the hydrogen bonds, meaning there is another unzipping or unwinding. So the helicase will go through the middle of DNA strand and will proceed to the unzipping number 2. Okay. So as mentioned earlier, the leading strand replicates continuously. Therefore, there is no need for the new primer to be attached in the leading strand. However, since the lagging strands run in an opposite direction, primer is needed to be attached for the polymerase, again for the polymerase 3 to build or construct another nucleotides. It is again because of its absences of hydroxyl group. So let's attach another primer here in the leading strand. And we may now proceed to the leading strand. So cytosine is paired by guanine. Let's always consider the direction of the replication, and then thymine to adenine and adenine to thymine. And let's go now to the lagging strands, which run from right to left, from 5' to 3' direction. So adenine is paired with thymine, and guanine is paired with cytosine. Now, as you observe here in the lagging strand, wait, let's separate. Okay, so here in our lagging strand, as you can see, there is a series of primer and a series of primer and nucleotides. Now, this is what we call the Okazaki fragments. It is because that is what I have mentioned earlier, that the lagging strand will replicate through segments, and that is what we call the Okazaki fragments. The primer, nucleotide, primer, and the nucleotides. Now, to finish the process of DNA replication, the DNA polymerase 1 will remove this primer. So here is our DNA polymerase 1. And will fill the gaps by putting the pairing of nucleotides, which is adenine to thymine and cytosine to guanine, and what is this? Thymine to adenine. Now, to form a continuous double strand, DNA ligase will seal and ensures that the nucleotides are attached in a DNA replication. Okay, so here is the DNA ligase. So it serves like a glue. It will ensure the attachment and the seal of these nucleotides on the strands. So as we can see here, we have now 4 strands. 2 strands for the old strands, which is represented by white zipper, and new strands, which is represented by the black zipper. So this is the byproduct of the semi-conservative mode of replication, that in every old strands, which is this white strand, there is a new strand, corresponding new strand. So I hope that I share what I have to share. I hope this explains the process of the DNA replications, and thank you for listening.